Okay, so welcome to this next video in which we are looking at the antithrombotic mechanisms of the endothelium. Okay, so we are currently revising the process of hemostasis so that we can understand how the antithrombotic mechanisms are going to work. So far we have seen platelet adhesion. The next portion we're going to see is platelet activation. Okay, so in platelet activation, what happens is that when the constituents of the blood move out through the hole in the blood vessel wall, then what they're going to come up against are loads of constituents of the blood vessel wall which they would not usually see. So for instance, they would not usually be exposed to collagen. Okay, but there is loads of collagen in the basement membrane here. There's then loads of collagen in the layer below that, the subendothelial connective tissue, which we previously showed in pink. Okay, we know then after that you have the internal elastic lamina in blue. Okay, and then after that you have the layer of smooth muscle cells, and again there'll be quite a bit of connective tissue in there. Okay, so basically platelets are going to see collagen for the first time when they're usually going around in the bloodstream. All they usually see is the constituents of the blood, okay, so the other constituents of the blood, and then the apical surfaces of endothelial cells, and they do not see collagen. So basically, when you have this hole in the side of the blood vessel, they are now going to see collagen, which is which makes up the more peripheral layers of the blood vessel wall. Okay, so collagen now is going to have an effect on these platelets. It's going to lead to activation of the platelets, and don't worry, I will explain to you what that means. So activation is going to occur. In addition, there is another thing which can drive activation, another very important molecule which can drive activation, and this is tissue factor, also known as factor 3. So tissue factor. Okay, and the old name for tissue factor is factor 3, and I'm telling you this not because people generally refer to tissue factor anymore as factor 3, but in case you're wondering why, when we later talk about the coagulation factors, you might wonder why there is no factor 3, and it's because it's so important that it's been given a new name, basically, which is tissue factor. Okay, now this is a protein, basically, that is on the surface of peripheral cells. So if we have a peripheral cell here, this will have tissue factor on its cell surface membrane. So here, this protein that I'm drawing on the surface of the cell, here, this is tissue factor in violet purple here. Okay, so peripheral cells in the body have tissue factor on their cell surface membrane. Okay, now the apical membrane of the endothelial cells does not have tissue factor within it. In addition, the cells that are within the bloodstream do not have tissue factor within it. So basically, platelets do not usually see tissue factor. However, when they go into this hole in the blood vessel wall, they're going to see loads of cells that make up the more peripheral portions of these of the blood vessel wall. So for instance, the smooth muscle cells outside the internal elastic lamina. Also the fibroblasts, which are responsible for producing all of this connective tissue, those will have tissue factor on their surface membranes, and this tissue factor is also going to lead to activation of the platelets. Okay, so let's draw a platelet here and let's see what happens when the platelet is activated. So here's our platelet, and I'll highlight it in orange again. Okay, now when a platelet is activated, what's going to happen is that it's going to start releasing a huge number of molecules, two of which we're going to look at. Okay, so the two molecules that we're going to be really interested in is it's going to release firstly ADP, adenosine diphosphate, and secondly thromboxane A2, which is often abbreviated to TXA2. So I assume you know what ADP is, but maybe you haven't come across thromboxane A2. You're probably less familiar, at least, with what it is, so I'll write out its name for you. Thromboxane A2. okay? Often abbreviated to TXA2. Now, let's start with ADP because its role is slightly simpler. ADP is going to trigger a chain reaction, basically. So, what happens is some of these platelets initially come into contact with collagen or tissue factor, okay? And this causes them to activate, and then they dump ADP and thromboxane A2 into the blood, basically, okay? Now, ADP is going to activate other platelets, so you don't need all platelets to directly meet collagen and tissue factor. Instead, only a few need to actually meet the collagen and tissue factor, and then they can set off a chain reaction that leads to the activation of the others. So basically, on the surface of platelets, so here is another platelet, you have a receptor for ADP. Okay, so we'll draw this here. So this is an ADP receptor here. ADP receptor. Okay, and what's going to happen is that when the ADP binds to the ADP receptor, that's going to trigger the activation of this second platelet. So this second platelet then is going to start releasing ADP and thromboxane A2. Okay, so then what's going to happen is the ADP is going to go off and activate more platelets. So what you can get is this positive feedback effect, or this chain reaction, where you start by activating a few platelets via them touching collagen or tissue factor, and then what it leads to is all of the platelets in that vicinity being activated. Now, that's great, but what's the point of it? Okay, well, the point of it is to produce this thromboxane A2. So the other component of what activated platelets are spitting out into the blood is the important bit. So you set off a chain reaction of platelets becoming active so that they can then produce loads of this thromboxane A2. So what's basically going to happen is in the vicinity of this cut in the arteriole, okay, so here's our cut in the arteriole, in the vicinity of this cut, you are going to get, and I'll draw the actual sides of the blood vessel, okay, so now it just looks a little bit odd actually because I need that removed. Okay, so imagine that I haven't got this little sharp bit in there because this is supposed to be the lumen of the blood vessel. In fact, let me draw this picture again, it won't take too long. So here is our cut in the side of the blood vessel wall. Okay, and there's the lumen of the blood vessel in the middle here. So this is the lumen of the blood vessel, and I'll just colour it in red to get the point across. Okay, so this is the lumen of the blood vessel where the blood should be, but obviously we've got this hole in the side, so blood is going to come out of there. Now, in the vicinity of this cut, what is going to happen is we are going to produce absolutely loads of this thromboxane A2. So let me denote thromboxane A2 by a turquoise dot. What you're going to get, basically, is thromboxane A2 absolutely everywhere, basically. Loads of this stuff, because we have set off a chain reaction of platelets producing thromboxane A2 and releasing it into the blood. Okay, now what does this thromboxane A2 do? Well, firstly, it causes vasoconstriction of the smooth muscle that lines the blood vessel. So it's going to act to cause vasoconstriction. So what thromboxane A2 will do is it will cause the smooth muscle cells that line the blood vessel to contract, okay? And when they contract, what will happen is their length will go down. And remember I told you that they assemble into these rings that surround the lumen of the blood vessel. So when all of the smooth muscle cells in one of those rings become shorter, then that means that the circumference of the entire ring is going to become smaller. And that means that the diameter of that ring is going to become smaller, and therefore it's going to constrict the lumen. So thromboxane A2 is going to lead to vasoconstriction. Okay, and this is a very important part of the hemostasis response. In fact, we say that hemostasis consists of 3 components, and vasoconstriction is the first component of the hemostasis response. Okay, so what's the point of it? Well, if you constrict the blood vessel, then less blood is going to be able to move through this blood vessel. So let me draw a picture of our constricted blood vessel. So this is going to go to something that looks like this. Okay, so really narrow lumen now, basically. So this is the blood vessel after it's vasoconstricted. And hopefully you can understand that less blood is going to be able to get through this constricted lumen to the point where you've got the hole. So you're going to get less blood loss, basically. You're going to get less hemorrhage, and I'm sorry, it's not even visible. There we go. So you can see that the lumen is hugely constricted now. So I'll draw the lumen in pink again. So this is now the lumen in the middle here. So the amount of blood you're going to be able to conduct down this passageway to the hole is hugely reduced. So hemorrhage is going to go down. So that's a major way in which you reduce hemorrhage. Okay, so that's the first component of the hemostatic response. Now, thromboxane A2 has another effect. It is going to cause the platelets to become sticky. So we've talked about platelet activation now. The final step in involving platelets is we need to get platelet aggregation, and this in fact is the second component of the hemostatic response. Okay, so platelet aggregation is 2. So what's going to happen is that platelets have released all of this thromboxane, okay, and we've talked about how this thromboxane has caused vasoconstriction. The other effect is that the thromboxane is actually going to act on the platelets themselves. So the platelets have a receptor for thromboxane on them. Okay, so I'll draw this here. It's actually a G protein coupled receptor, so I might even draw it as a 7 transmembrane receptor. There we go. Okay, so this is the thromboxane A2 receptor, TXA2 receptor. Okay, and when thromboxane binds to this thromboxane A2 receptor, so we've got all these platelets that are gushing this thromboxane A2 out, what's going to happen is that when the thromboxane binds to the thromboxane A2 receptor on a platelet, it's going to lead to that platelet becoming sticky. Okay, so it leads to an alteration of another protein on the surface of the platelet. So let me show this protein here. So this protein is known as glycoprotein 2B/2A. Okay, so 2B/2A. Okay, and this is going to be very important for platelet aggregation. So when thromboxane binds to the thromboxane A2 receptor, the thromboxane A2 receptor leads to an activation of the glycoprotein 2B/2A protein, and this now is going to become capable of binding to fibrinogen. Okay, so let me now describe the next process in this, which is platelet aggregation. Okay, so what basically is going to happen is that you are going to get platelets aggregating together, and this is how. So when the thromboxane A2 has bound to the thromboxane A2 receptor on the platelets, it leads to a change in the glycoprotein 2B/2A protein, so GP2B/2A here, and now GP2B/2A is capable of binding to a protein known as fibrinogen. Now, we are going to see fibrinogen later, so we might as well have a little bit of a discussion of it now, because fibrinogen is extremely important in the coagulation cascade. Okay, so fibrinogen is also known as factor 1. And again, no one will ever call fibrinogen factor 1. Everyone calls it fibrinogen now. However, just so that you know what factor 1 is and why there is no clotting factor 1, well, the reason that no one ever talks about clotting factor 1 is that it's so important that it's had a new name given to it, which is fibrinogen. Okay, so fibrinogen has this bilobed structure. It has 2 lobes connected by a protein linker between the 2. And what can happen is that this GP2B/2A, once it's been activated by thromboxane A2, it's now capable of binding to one of the lobes of fibrinogen. Okay, So here is a lobe of fibrinogen, like so. So it will bind to one lobe, so now the platelet is linked to a lobe of fibrinogen. Now, if you imagine having another platelet here, okay, so here's another platelet, which has also been activated by thromboxane A2, so that its GP2B/2A protein is capable of binding to fibrinogen's lobe, or one of the lobes of fibrinogen. Then you can imagine that you're going to get this link between the two platelets. Now that's very nice, but how is this going to actually lead to a huge aggregation of platelets? Well, the reality is, do you think each platelet just has one GP2B/2A? Of course, the answer is no. Each platelet will have multiple GP2B/2A proteins, and when the thromboxane A2 activates the platelet by binding to its thromboxane A2 receptor, then it's going to lead to the activation of all of these GP2B/2A proteins, so that they're all now capable of binding to lobes of fibrinogen. And I should say, fibrinogen is a normal component within the blood, so it's a protein that is within the blood. It's a clotting factor. It's the zymogen. Okay, so it's the zymogen. So it's an inactive protein that's going to become an active protein in the coagulation cascade. However, here is another use of fibrinogen other than activating it to fibrin. Okay, so you don't need to activate fibrinogen for it to perform this role. All you need to do is activate the GP2B/2A proteins on the surface of the platelets. Now, what you can imagine is you're going to form a great lattice now, because if we bring more platelets in here, and these platelets too have been activated by the thromboxane A2, okay, and I shouldn't use that word activated, because platelet activation means that reaction where they begin to release the thromboxane A2 and the ADP, and it's triggered by, well, the initial ones are triggered by collagen or tissue factor, but then later it's triggered by ADP binding to the ADP receptor. Okay, that, strictly speaking, is what is meant by platelet activation. Okay, so I shouldn't really say that thromboxane A2 activates the platelets, because it doesn't. It activates the GP2B/2A so that aggregation can now occur. It activates platelet aggregation, really. Okay, so you can imagine that you can continue this picture on, and you're going to get platelet linked to platelet linked to platelet linked to platelet. So you're going to get a massive, great aggregate of platelets. Okay, now, in the next video, what we'll do is start to piece this all together and see how we're going to get a mass of platelets blocking that hole in the side of our blood vessel.